Hi guys, welcome back to again Adya online biotechnology classes. Today I will discuss about deamination reaction. So let me know about what is a deamination. Deamination is nothing but the removal of amine group from the amino acid cell, liberation of ammonia to form keto acid. That is known as deamination reaction. Okay, so let me understand what is the deamination reaction. If you take the amino acid like glutamate, so glutamate consists of three hydrogen molecules, and these three hydrogen molecules, the two hydrogen molecules are liberated out by the addition of NADP, and these NADP it gives positive charge. So these NADP it accepts the hydrogen molecule, gets converted into NADPH and H plus ion. Okay, are you understand? So deamination is nothing but removal of amine group, from the amino acid cell, liberation of ammonia to form keto acid is known as deamination reaction. Okay, what is deamination reaction? The removal of amine group from the amino acid cell, liberation of ammonia to form keto acid is known as deamination reaction. Okay, so normally if you take the amino acid like glutamate, glutamate consists of three hydrogen molecule, and from that three, so how to draw the amino acid? First of all, so this is the carbon atom, it is the central carbon, it consists of carboxyl group, and another one is the amine group, and another one is the hydrogen molecule, and this is the R group. R is nothing but side chain. Okay, R is nothing but side chain. This is the structure of amino acid. Okay, so amino acids consist of three hydrogen molecule. So normally if you take the amino acid like glutamate consists of three hydrogen molecule, and these the two hydrogen molecules are liberated by the addition of NADP, and these NADP will accept the hydrogen molecule. From that hydrogen molecule gets converted into NADPH and H plus ion. So the two hydrogen molecules are liberated out. So this glutamate gets converted into alpha aminoglutamate. Why? Because the two hydrogen molecules are liberated out. Okay, so the carbon, so the functional group is attached to the central carbon, so this carbon is also called the alpha amino position or alpha aminoglutamate. So this glutamate gets converted into alpha aminoglutamate, and this alpha aminoglutamate, by adding of water molecule, the water molecule is very necessary. So the structure of water molecule, oxygen and hydrogen, another hydrogen, H2O. So the water molecule is very necessary. So the water molecule comes and binds with the NH position. Okay, NH is nothing but imino. Okay, imino. So NH is nothing but imino. NH2 is nothing but, okay, NH2 is nothing but amino. Okay, amino. So the water molecule comes and binds with the NH position. So the how many number of hydrogen molecule? The three hydrogen molecules. It consists of three hydrogen molecule. So in the reaction, the NH3, the liberation of ammonia is liberated out. Okay. So the alpha aminoglutamate, by adding of water molecule, the water molecule is very necessary, and the water molecule comes and binds with the NH position and liberation of ammonia. So this oxygen, it will be act as nucleophile. So this oxygen comes and binds with the carbon in double bond in nature. Okay, double bond in nature. So these are nothing but keto acid. Okay, keto acid. So these are deamination reaction. Okay, so are you understood? So what is deamination? The removal of amine group from the amino acid cell, liberation of ammonia to form keto acid is known as deamination reaction. Normally if you take the amino acid glutamate consists of three hydrogen molecule. From that the two hydrogen molecules are liberated by the addition of NADP. NADP, it gives positive charge. So this NADP, it accepts the hydrogen molecule, gets converted into NADPH and H plus ion. So this glutamate gets converted into alpha aminoglutamate. Imino is nothing but NH position. Why? Because the two hydrogen molecules are liberated out. Okay, so these alpha, so why we call it as alpha position? So the functional group is very near to the central carbon, that is known as alpha aminoglutamate. So these alpha aminoglutamate, by adding of water molecule, so water molecule, it is very necessary. The water molecule, the structure of water molecule, okay, oxygen, hydrogen and hydrogen, H2O molecule. So the water molecule is very necessary. The water molecule comes and binds with the NH position. Okay, so what will happen? So the water molecule comes and binds with the NH position. So already NH position, how many number of hydrogen molecule? One hydrogen molecules will be there. So the water molecule consists of two hydrogen molecule. So these two hydrogen molecules comes and binds with the another hydrogen molecule by the liberation of NH3. NH3, okay now, how many number of hydrogen atoms? 1, 2, 3, okay, so 3 hydrogen molecule, it is liberated of ammonia, okay, to form keto acid, okay. So this carbon is double bond in nature of the oxygen, so remaining is from oxygen. So this oxygen, it will be act as nucleophile. So this oxygen comes and binds with the carbon position, so it will be converted into keto acid, is known as alpha ketoglutarate. So these are the deamination reaction. So this deamination reaction consists of two types. One is the oxidative deamination and another one is the non-oxidative deamination. What is oxidative deamination? What is oxidation and what is reduction? Oxidation is nothing but the addition of oxygen and removal of hydrogen. That is known as oxidation. What is the reduction? Addition of hydrogen and removal of oxygen. That is known as reduction reaction. So this deamination, it consists of two types. One is the oxidative deamination and non-oxidative deamination. So let me know about what is the oxidative deamination. So oxidative deamination, it consists of two types of amino acids will be there. One is the levoamino acid and dextroamino acid. Okay, levoamino acid and dextroamino acid. So these levoamino acid in the presence of protein, flavoprotein, that is nothing but flavin mononucleotide. Next, dextroamino acid, it undergoes FAD. FAD is nothing but flavin adenine dinucleotide. So both are flavoproteins. Okay now. So how to will convert the levoamino acid gets converted into keto acid and dextroamino acid gets converted into keto acid. So first of all, the levoamino acid gets converted into alpha ketoglutarate acid, okay now, by adding of water molecule and liberation of ammonia, okay now, to form keto acid in the presence of enzyme levoamino acid oxidase. Okay, so the deamination consists of two types. One is the oxidative deamination and non-oxidative deamination. In this oxidative deamination consists of two types of amino acids, levoamino acid and dextroamino acid. So the levoamino acid, it consists of 3 hydrogen molecules will be there. So and these 3 hydrogen molecules, the 2 hydrogen molecules are liberated. So 2 hydrogen molecules are liberated by the addition of NADP plus. So this NADP, it accepts the hydrogen molecule, gets converted into NADPH plus H plus ion. Okay, NADP, it accepts the 2 hydrogen molecule, gets converted into NADPH plus H plus ions. Okay, so next the levoamino acid, by adding of water molecule, the water molecule is very necessary. The water molecule comes and binds with the levo position or levoamino acid by the liberation of ammonia in the same reaction. Okay, so in the presence of enzyme levo amino acid oxidase, so gets converted into alpha ketoglutarate acid. So next one is the dextro amino acid. So dextro amino acid gets converted into alpha ketoglutarate. So these dextro amino acid having 3 hydrogen molecule. From that the 3 hydrogen molecule, the 2 hydrogen molecules are liberated out. So the dextro amino acid in the presence of enzyme dextro amino acid oxidase. So these dextro amino acid will remove the 2 hydrogen molecule by the addition of NADP plus. So this NADP will accept the 2 hydrogen molecule, gets converted into NADPH plus H plus ions. Okay. So it undergoes the it takes place the enzyme dextro amino acid oxidase. So in this reaction levo amino acid oxidase enzyme. So in this reaction dextro amino acid oxidase enzyme. So by adding of water molecule, the water molecule is very necessary. The water molecule comes and binds with the NH position, same reaction, NH position. So by the liberation of ammonia, okay. So the hydrogen molecules are the 3 hydrogen molecules will be there. So the liberation of ammonia, NH3, liberation of ammonia to form keto acid. So these carbon is double bond in nature of the oxygen. So these are nothing but keto acid. Okay now. So next one is the non-oxidative deamination. Non-oxidative deamination consists of 2 types of enzyme. One is the dehydrase enzymes and another one is the desulfidase enzymes. So first of all dehydrases. So dehydrase enzyme. So if you take the amino acid like serine, okay. So serine consists of 3 hydrogen molecules will be there. From that the 2 hydrogen molecules are liberated out. So by the addition of NADP it accepts the hydrogen molecule NADPH and H plus ion. By adding of water molecule, liberation of ammonia to form pyruvic acid. Why? Because in this reaction, in the final product will be formed alpha ketoglutarate acid, and in this reaction the final product will be formed pyruvic acid. So why? Because in the presence of oxygen aerobic respiration, in the absence of oxygen it is nothing but reduction reaction. So the final product will be formed pyruvic acid. Is it right? Okay. So next one is the dehydrases. So normally if you take the amino acid like serine consists of 3 hydrogen molecule. From that the 3 hydrogen molecule, the 2 hydrogen molecules are eliminated by the addition of NADP. NADP gets converted into NADPH and H plus ion by adding of water molecule from the NH position by the liberation of ammonia to form pyruvic acid. Okay now. So next one is in this reaction it takes place by the enzyme dehydrase enzyme. Next another example, so cysteine. So normally if you take the amino acid like cysteine, so cysteine is a sulfur containing amino acid. So this cysteine consists of 3 hydrogen molecules will be there. From that the 2 hydrogen molecules are liberated by the addition of NADP, and this NADP will accept the hydrogen molecule, gets converted into NADPH and H plus ion by the adding of water molecule, liberation of ammonia to form pyruvic acid. Okay, another example of the non-oxidative deamination. So in this overall deamination reaction, so it is led by the which type of enzyme? Alpha ketoglutarate acid. So So in this deamination reaction takes place by the enzyme glutamate dehydrogenase enzyme. So this glutamate dehydrogenase enzyme is present in the mitochondrial matrix. It is made up of zinc. Okay, it is made up of zinc. So these glutamate dehydrogenase enzyme, it is regulated by the adenosine triphosphate, adenosine diphosphate, steroid hormones, steroid hormones and thyroid hormones. Okay, thyroid hormones also. Next, this glutamate dehydrogenase enzyme, it is activated by the adenosine triphosphate and guanosine triphosphate. Guanosine triphosphate. So overall of this deamination reaction, what is the deamination? The removal of amine group from the amino acid cell, liberation of ammonia to form keto acid is known as deamination reaction. So normally if you take the amino acid like glutamate consists of 3 hydrogen molecules. From that, the 3 hydrogen molecule, the 2 hydrogen molecules are liberated by the addition of NADP, and these NADP it accept the hydrogen molecules, gets converted into NADPH and H plus ion. So these glutamate gets converted into alpha aminoglutamate. So why we should call it as alpha aminoglutamate? So the removal of 2 hydrogen molecules. So the 2 hydrogen molecules will be removed. So and again NH position. Okay. So this alpha aminoglutamate. So this alpha aminoglutamate glutamate, by adding of water molecule. Water molecule is very necessary. The water molecule comes and binds with the NH position, and water molecule, the structure of water molecule, this is reaction. So the water molecule comes and binds with the NH position, the liberation of ammonia to form keto acid. So this oxygen is, it will be act as nucleophile, comes and binds with the carbon in double bond in nature. So it is nothing but keto acid. Okay, so these are the deamination reaction. So in this deamination reaction consists of 2 types. One is the oxidative deamination and non-oxidative deamination. Oxidative deamination, it is takes place by the in the presence of 2 types of amino acids will be there. One is the dextroamino acid and levoamino acid. So non-oxidative deamination consists of 2 types of enzymes will be there. One is the dehydrases and desulfidrases. Okay, so in this reaction, so which type of enzyme is participated into the deamination reaction? Glutamate dehydrogenase enzyme. So this glutamate dehydrogenase enzyme, it is only found in the mitochondrial matrix and it is made up of gene. So this glutamate dehydrogenase enzyme, it is activated by the, it is regulated by the allosteric reaction. That means the adenosine triphosphate, guanosine, adenosine diphosphate, steroid hormone and thyroid hormone. It is activated by the adenosine triphosphate and guanosine triphosphate. Okay, thank you.